feel free to interrupt at any time. And just wanted to point out that since there was a question on Pearson correlation, I did post a very simple thing on Pearson correlation. So if you like simple explanations, here's one. If you do ever have some suggested things that you think might be helpful for other people, let me know and I'll post them on the schedule page as well. So there's a lot of different things that is useful to know about data analysis. If you find a good website or a good paper source, please do email me and I will post. If we do, I have not yet set up any sort of wiki page for the class. Let me know if you would like one, and if there's a particular format that we could do, a Google group, a wiki, there's all sorts of different things we can do. Let me know if you have a preference. So my apologies for going quickly through the Mapper software, but I believe that for the workshop, we will be focusing more on persistent homology type stuff. So we're only spending this 1 week on Mapper. There's a whole lot more to say. So I'm going to, since I didn't quite finish what we were discussing last time, I'm going to quickly finish off the slides for what we did last time, and then we will demonstrate one version of the Mapper software. There are many versions of the Mapper software. So in terms of, remember we were looking last time at breast cancer gene expression with L infinity centrality, just to point out that we are looking at how far things are from the norm, and so this works well for breast cancer data or cancer data in general because your tumor cells are probably far from the norm, which is what's causing the problem. Normal things might be okay. And we did have a second filter as well and used single linkage clustering. It was microarray data, which meant that our data points were various different patients. So our data points are various different patients. So this would be a vector if we go straight across here. That was assuming I could actually draw straight lines. That would be a single vector. And then the elements of our vector are having to do whether the gene is upregulated or downregulated in comparison to a comparison group. And so you just get a vector of numbers saying whether it's upregulated or downregulated. And the data set actually contains about 24,000. If you want to actually look at the data set, you can download the data set and look at it yourself. There'll be some README files and several different data sets within here. And that's for this, the first paper is actually the first one on the list, but they list other places you can also get data. And there's a variety of places where one can get this data, or one can get data. This data is posted at NKI. So we do have a vector that's 24,000 long for the NKI data set. But if you take a look at all the 24,000 data set, you just get, something that looks pretty much like this. Ignore the coloring for now. So at the moment, we're just looking to see how it clusters, and it clusters into this one thing. It is flared out does mean that there should be a difference from this end to this end. So we are getting more information than just clustering. The flares do tell us that they're, the graph themselves does give us some information. But the problem is we've got 24,000 genes, and so today I'm on my Mac, and so my writing instrument is my finger, which does not work well. But suppose we have the vector 1, 2, 3, and then maybe all the other values are all 1. So maybe we have all 1 for all the other 24,000 minus 3 that I've written up here. And then maybe for the second patient, maybe it's 3, 2. So 3, 2, 1, and then everything else is a bunch of 1s. Well, if you calculate the distance between vectors where the only difference between them are in the first 3 components, but everything else is the same, their distance won't be that great. So you don't want to be comparing all 24,000 genes because many of the genes are just acting normally. And so they're not going to give you any useful information. In fact, they will be hiding the useful information. And so thus, one works instead with the top most varying genes. And so you can see if you set your threshold, you look at all the genes, there's not too much there is some variation. We do get this nice line here. With 11,000, it's pretty much the same. If we take a look at the top 7,000 most varying genes, we start getting things splitting off here. And the top 15,000 things split off, slightly more. And so they took a look at the top 1,500, not 15,000, but the top 1,500 is basically where they decided to set the threshold. Frequently, when you're looking at these things, they're trying to find the top 70 most varying genes if you use the more, not the topological data analysis, but you're trying to find not that many genes. The nice thing about topological data analysis is, yeah, you still want to use a threshold, but the threshold, whether we set it at 3,000 or 1,500 or 1,000, the results probably won't vary too much. Versus many other types of data analysis, they're trying to find, the top 70, the top 86 or something like that, which is quite a bit different. Now, you still might lose some information. We are only looking at the top 1,500 genes in this paper, in their paper, but it is that way we don't hide other things. But that does mean that certain things might be harder to find, so you still might lose certain types of genes. And again, if you've got questions, feel free to interrupt at any time. Okay. So that is one of their things. They just also noticed that they did say that comparison was, so this was a quote from this paper that was published in The Lancet in 2005, that they found it difficult to compare results because of the different techniques. And they were trying to find the top, fewer than 100 genes. Versus, if you now take a look at the results, and the results which we quickly looked at the end of class last time is over on the left here. So look just on the left, and again, ignore color for the moment. We can see that between the two data sets, among the survival group, there is a lot of similarity between the two data sets. So even though they were very different data sets done on different platforms, we're still getting somewhat similar pictures, at least for the survival. When I say survival, remember in one case we had survival versus death. In the other case, it was relapse versus no relapse, which means they probably survived, versus relapsed, which is not a good thing. They are more likely to be in the not so good death group. So the partitioning is not identical, but there should be a strong correlation between no relapse and survival. And we can see that the output is very similar. Not identical, this red arm here is much smaller than this arm right here. It's shorter, but there is still quite a bit of similarity. There is, more difference in the relapse group, so there is some differences, but we can still compare them fairly nicely and fairly visually, which is useful. So any questions about the output before we add color? Well, in terms of the adding color, on the left again, it's colored by ESR1 levels. It is known in the breast cancer community that if you have high ESR1, that means that you are likely to survive. So a high ESR1, you get that result, you say good, that means you're likely to survive. But not always. You can see that there are exceptions to that rule. If you're in the low ESR group, you're not too happy because you can see that there's a lot more blue up here. A lot more blue. Some, red, so some high ESR within here, but some low ESR here. So low ESR ones are less likely to survive than high ESR ones. But we can see again here that there is a low ESR1 group that did survive. And so it is not a perfect correlation, and doctors and patients would like to understand that better to know what group you are more likely to be in. And so we can then study these two groups. So it was the authors of this paper decided to then study the low ESR ones that survived apart from the low ESR ones that did not survive. And in doing so, they then said, well, now let's take a look at what's the difference between this group and this group. So the low ESR1 within the survival group versus the low ESR1 groups in those who did not survive. And so they compared the two and found that if you color by chemokine levels, you can see that that somewhat differentiates out between those who those who did, survive. There was a higher chemokine activity, which is related to the inflammation pathway. And so this is redder than this. Yeah, there's still some red, so it's not a perfect sort of stuff, but they did find some things that did distinguish between the two groups. So when you do this data analysis, the first thing you do is you just apply your software and get your results. But once you have your results, it's helpful to be familiar with the applications. You can explore it more to say what's the biology behind that, and can that determine how we affect the treatment. Let's take a closer look at this sort of subgroup, the low ESR1, and see if we could have, so if we take a look closer at this tail, what if we use other data analysis techniques? Well, if you use other data analysis techniques, then that group gets clustered among a variety of different, so it gets clustered among a variety of different, let's go back. It gets clustered. You can't really see that from clustering. It gets divided, and same thing with PCA. So finding this blue tail here is hard to find using other methods, that via the topological methods. Finding this blue tail, you can just see it so nicely. So that finishes up our last lecture. Now let's go ahead and discuss the Mapper software. So to actually apply the Mapper software yourself, you've got a lot of choices. One of the things is that there is a web resource. So if you go to comptop.stanford.edu/pad, you can actually do all your analysis via the web. But there are, when you've got web resources, there are restrictions. This is set up specifically to do disease-specific genomic analysis. You can't do things with just Mapper, but you do have to compare a normal data group to a tumor data group. So this is set up specifically for that. So if you want to do something more general, you probably don't want to do the web interface. But if you want to do specifically comparing tumor data to normal data, then it's set up specifically for that. Just a couple more words about PAD. PAD basically creates this healthy state model. So they take an average. So if we take a look at this paper right here where it's described in PNAS, they create this healthy state model. Then you take the vector. So remember the vector that was created by taking a look at all these genes here. We can take this vector here and project it onto the healthy state model and then take the difference. So you always want to know the difference between your tumor versus your healthy. And so when they do the distance, they're talking about the length of this vector. So this is the vector that they will then analyze. When I say the length of the vector, I might mean standard Euclidean length. In actuality, you often mean LP length. So instead of taking the square of each component, adding it up, and taking the square root, you might take the cube of each component, add it up, and take the cube root if you want to do the L3 norm. There's a lot of different distances that are involved. But once you have this vector, you can then choose what distance to use it. And I believe they used the L-form norm and then took it to the second power for their distance. And just so that you've seen it, this is actually the result that we get. And so this is the result that we see right here using this other method. So they're also comparing breast cancer data, but they're using a different norm, a different filter, and using using this filter right here with this vector here and that norm, they get a different result. I would say, again, analyze the biology and come up with some good conclusions that give different information. So remember, there's lots of different ways one can explore the data. I'm hoping that maybe near the end of the semester we might compare the various different methods that we will go through. So, okay, so that is one version. Then there is also the PythonMapper. PythonMapper is probably the one I should have demonstrated because it does have probably the best documentation. But I have to learn Python first before I demonstrate it. And so if you're interested, it is something I do plan on playing around with this semester, but not today. But it does have some nice documentation. If I scroll down to the next page, you can see that it does have some built-in filter functions. And one of the nice things about Python is it is compatible with other programs. So if you're familiar with R, maybe you want to do your clustering in R. There's a lot of modules for calculating distances, for applying your filters, for applying your clustering. And so you can use R subroutines in addition to the Python if you want more filters than what are plugged in here. But they do have several different filters plugged in into the Python Mapper. So if you go to this website, you can see the various different filters that are plugged in. Then there is, I don't actually know how to pronounce it, IASTI, anyone know how to pronounce it? So this is proof that you can actually make money in mathematics. So Gunnar Carlson and his group, among others, started this company called IASTI. My apologies for my pronunciation, and they have their own software, so you can pay to get your data analyzed, or you can do it yourself with their software here, because there is a academic free trial. Their academic free trial, however, you have to apply. You may not get accepted, and you do have to include, I think it was a one-page proposal describing what you're doing, and then you will get a 3-month free trial. But it is something worth trying out, and if you do go to the IMA workshops, you can certainly talk to him more about what his software can do. But that is also an option. What we're going to take a look at is the MATLAB version, and so we're going to go ahead and see if we can do a live demonstration, and so we will see if that works. So if I go to, I've got it set up in different spots. So that is if you downloaded the Mapper software, if you clicked on that, you can go ahead and do that. I'm going to demonstrate it on the Linux platform. You can also run it on a PC or a Mac, whichever you prefer. But basically, I just opened it up in MATLAB. So what I opened up was the Mapper tutorial. So if you go ahead and download, I'm going to show you where it's. So I downloaded, extracted it. It got me all these .m files. And then there is a brief tutorial in an HTML document that you can also take a look at. I thought I actually tried it on a PC, but no, I tried it on my Mac. So yeah, you will have to untar it someplace else unless you, there are ways of untarring it on a PC, but you have to have the appropriate software, which most PC people, unless they do a lot of other stuff, might not have. If you want the untarred version on a PC, go ahead and email me and I can send it to you. These are not large files. These are actually very small files. So it would be very easy to get via email. So we can see that there are just a few files within here, and they are all actually fairly short. If you click on it and it's set the mapper tutorial.m, you can then open it up in MATLAB. And when we open it up in MATLAB, we get this thing here, which you can then run. Before I run it, however, in this thing here, so we have a, actually I did put this on, oh no, I was going to run it from my, never mind. So because I'm running it not on my Mac, because I don't have the thing, that I don't have MATLAB installed on my Mac, I'm using a virtual desktop. So I thought it might be safer, plus I can more easily change the font if I actually go over and run it on a terminal. So I'm going to do the terminal version first just to illustrate a few of the things. And so I copied a few things over here just to say what the first few lines are, and then we'll look at the HTML document, and then we'll go back and look at the actual stuff here. So the first thing is there are a number of MATLAB commands that you actually don't need for Mapper, but for our illustrated purposes, I'll go ahead and demonstrate them. I'm going to change the random from 300 to 2, so I'm doing random 3, 2. Oh, and then I'm going to get rid of the semicolon so that I can actually see the output and hit return. And you can see that I got 3 data points that are each having a vector of length 2. So if I wanted to get 300 points instead, I can do 300. But since I don't want to see a matrix that's 300 by 2, I will suppress the output, and I will do that instead. Let me go ahead and get back to the 3, 2 so that we can see a few more things. Then the next thing that they did is they just divided each coordinate by the norm. So if you take vector 1 and divide by its norm, you create a unit vector. And so now, actually, let me get rid of suppress. So now when you see the output, these are now all unit vectors, so they will lie on the unit circle. So now our data set, we know what our data set is. It's nice sometimes to create a data set that you understand, because then you know what you expect the output to be. So you can test to see if this thing is working well. And so we've created a data set of points on the circle. Hopefully you kept your 300 points, because with 3 points we're not going to see much. I just wanted you to be able to see the output. The next thing is our L2 distance. L2 distance is in your MATLAB file. It was actually a program that had existed for a while, so it wasn't actually created for Mapper. It's basically just calculating the Euclidean distance between the 2 points. If you prefer something else, find another MATLAB function. There's going to be lots of different MATLAB functions that will calculate the LP norm. And so here we just calculate the L2 distance. And so because I only had 3 points, I get a 3 by 3 matrix that gives me the distance between every pair of points. And you can do it with the 300. Let's now, so that was basically just to illustrate a few things on there. Let me now get back to the HTML. So this is the HTML version, which basically just shows you exactly what's in that .m file and goes through that. So we generated the circle in 2 dimensions. We found the distance matrix. And now we're going to choose a filter. And so for the filter, they use the distance from the first point. And so this first line here is what gets us our filter. The second 2 lines are actually not needed. They are just if you want to see the output. So that's standard MATLAB stuff, to go ahead and do a scatter plot of our vectors using circles of a particular radius with our filter with equal axes. And so this one's a little bit small here, but you can see we just get out this circle here, a nice circle. So that is what our data looks like. and it was colored according to the distance from the first point. So you might actually have a different coloring. You can see that my first point is, it was in terms of, yeah, so my first point, I think blue is low, so my first point would be somewhere around the blue point. And then as you go around the circle, you get further and further away from that point. So if you want to see it, you can use standard MATLAB stuff to see what your data looks like before you do anything to it. Assuming your data is nice and two-dimensional or three-dimensional, it might be harder to see if you work with real data. But we can see our data here. We now can set our parameters for MATLAB. There are a few parameters one can set. Two of those few include the filter samples and the overlap percent. So in the overlap percent, remember we create intervals, and those intervals we want to overlap. And so, so we might have them overlap by 50%. And then maybe we create 5 intervals, maybe we create more, but let's actually take a look at that. I did have a couple of stuff here. So if you want to take a look at the L2 distance function, you can see that it is just creating the standard Euclidean norm, and it tells you how it's done. So all of these things are actually included within the mapper. If you read the if you open the .m file in some text editor, it is very nicely commented. In the beginning, they describe exactly what what it does, who created it, when it was created. And it also says someplace that you are free to modify, extend, and distribute. So I can email this to you without a problem, and you can modify and extend it as you wish. And that's the same thing if I go ahead and look at the actual mapper software. It again also has a nice documentation saying what the inputs are. And so we've got our default overlap is 50% if we don't specify it. Our default, so in terms of the length of our intervals, if we take a look at the length of our intervals, we will take our maximum minus our minimum and then divide by the number of intervals that we want. And so that's what we get. And so you can read over. It's a little hard to read over mine because I blew it up so the lines go off onto the next step and the text is still a little bit small. But you can, again, modify, extend, or distribute this thing here. And if you do take a look at, the margin has to do with the clustering, which is also described a bit in terms of here. I think I might let you look at it instead of scrolling up because I don't think the scrolling up works too well when one is working remotely on, using a virtual thing here and then a virtual thing there. So, but if I did scroll through, I will just scroll through quickly. You can see that the code starts about here and it ends, about there, and then the rest is commented out. So again, it's a very short code. So if you did want to, for example, do it in R or Mathematica, porting it to someplace else is probably not too difficult. But you can also, if you prefer R, you probably prefer the Python one anyway, possibly. I don't know. But you might prefer Mathematica or something else. So that's the code. Then we will run Mapper. So we just run Mapper. I'll go back to the MATLAB soon. And this is where, when you run the Mapper, that's what the Mapper does, is that it will then filter, so it shows you the different ranges for which it's filtering in. Then it's finished, and we are now finished with our Mapper. Except we would like to see the output. If you would like to see the output, they use GraphViz, which is a standard tool for visualizing graphs. And so now the rest of the things are, again, MATLAB and GraphViz commands. They are not actually Mapper commands anymore. So however you want to visualize is up to you. And so we label a few things, generate the input. Now, when you see my MATLAB code, you will notice that I removed the /temp. So I removed this thing because otherwise it goes into this temp file that I don't have easy access to. And so I'd rather it go into my home directory. And so I put it into either I remove the / so it doesn't go up a directory into some place that's not really my home directory. And I created a temp file, or I just created a mapper file where I put it in. So you'll see that I've actually removed this /temp in mine. So if you want to actually see the output, you might want to remove it or say wherever you want to put the output. And that you will see is in a variety of different places. So if I go down, not there, but it is in other places, you can see it's in a variety of places. So if you do one of them, you should change all of them. The other thing that you will have, that you might notice that I will have changed is this system command. So the system command is a MATLAB command that basically says we're now going to call another program outside of MATLAB. So we're now going to your system. And if we go to your system, I was able to delete this and didn't need the sprintf command, but we're basically printing this command to our system. You don't normally need that, but in case you do, you can leave that in. Maybe it was needed in Mac. You can see that they ran it on a Mac. So if you run it on a Mac and your GraphViz is installed in the same place as their GraphViz, then this command should work for you. But when I'm running it on a Linux box, my Nito, so Nito is this Nito right here, that is a GraphViz command. My Nito is located elsewhere. So there's two ways to access that Nito. One is just to remove all of this stuff. So if you remove everything before the Nito, there's a good chance, if your paths are set up, that it'll work without saying what the path is. If your path is not set up, then you can do a which Nito, and the which Nito will tell you. So I'm assuming you're on a Linux or on a terminal on a Mac. And so either way, if you get out a terminal on either a Linux or a Mac and you do which Nito, then it will tell you where the Nito is, and you can just replace this stuff with that instead. So make sure you have this / here as well in front of it. So when I do which Nito, mine said /usr /bin, and so then I did / the rest of it. So let's now get out of this and into the no machine that has probably gone to sleep. Okay, so to take a look at where I have it now, you can see in terms of the modifications, I did write straight to my directory. So I changed I removed all my //temps /. And then the other thing that I removed is my screen is changed resolutions when I plugged in to show the stuff to the Classroom at UI, and so I didn't notice until now that I don't have complete screen resolution. I actually don't see my entire screen. But fortunately, I can still see the one line that's of interest. You can see that I changed it. I removed the sprintf because it seemed to work just fine without it, but you can keep it in, and it should work fine with that as well. And I could go ahead and put in the /usr /bin the Nito, and that will find the Nito. Unfortunately, we seem to be having a problem with the Nito command on our Linux boxes, and we were not able to figure out why. It said it couldn't find something, but when we did a where is and stuff commands, it was actually where it was looking for it. And so I changed instead to dot. So let me show you what happened when I put Nito in instead. So if I put Nito in and now run the program, I then have another screen up here. I unfortunately forgot that I'd also unsuppressed some things in my file. So let me go back and suppress a few things. I don't think we need to see our 300 data points. So I think that was it. So let me run. So we'll try running it again and seeing that you can see that we did run our MATLAB. And then I get warning could not load. So we're having a problem with this plugin. Hopefully it will work for you. But what it does tell me is it does use one of circo.fdp. GraphViz has a lot of different ways of displaying a graph. I mean, basically, Mapper created this dot file that told them what are the vertices, how to label the vertices, how to, what are the edges between the vertices. So it gave that dot file basically described the graph. Now we just want to visualize the graph within GraphViz, and there's lots of different ways of doing it. And because my Nito didn't work, I got a list of ways to do it, circo.fdp, etc., lots of different ways of visualizing it. And so I tried dot. And so if I go back here and change this thing to dot, so change instead of Nito, so execute GraphViz, instead of Nito, I will put dot, and we'll save, okay. And then we'll go ahead and run it. And so now when I take a look, there should be, except for I think I actually might have, so I didn't, that is showing. So hopefully, there it is. And so now we've got the circle. If you do Nito, I believe, so Nito does an energy minimizing. Where you put the vertices so that the graph looks nice is a big computational problem in and of itself. Now, we have a simple graph, so basically whatever we do, we will be able to see that we basically have a circle here. Well, with our clustering, we got this little edge out here instead. So for us, with this very simplified data, it didn't really matter which program that we used. So with Nito, it would have been more circular because Nito does an energy where the vertices will sort of repel each other. But with dot, we just got a nice, pretty graph where we could still see that because we had a circle, we had some clustering going on here, and that is basically what our stuff looks like. Any questions in terms of getting this thing to work? So everybody able to get things to work, hopefully? Okay. If we want to look at additional sort of data, I can go ahead. I'm on the wrong computer. Let me go back and get out of this here. So the tutorial gives you a basic idea as to how the mapper works, but do note that many of the commands were actually, very few of the commands were actually mapper commands. Most of them were actually MATLAB commands that you get to choose. You get to choose using MATLAB how to calculate the distance. There is a default thing built in, which you're welcome to use, the Euclidean distance. There's a default clustering mechanism. There's a default way of visualizing using GraphViz. But if you do prefer other things, you can do so. So I think we can go ahead and now get into this. Okay. So now if I go into my downloads and see if I can check. I forgot to get out a file, so now you get to see my whole list of files here because I forgot to open them up. And the name of the file I'm looking for, maybe I will just re-download it because I have a horrible memory of where this thing was. I know what I can do. Is I opened the data recently in Excel, so I will just reopen Excel. So what I'm now going to take a look at is the array, the data from that NKI database. So if you download that database right here, you can see pretty much, let me see if I, I'll try 200% or let's try 300% and see if, yeah, that should hopefully be at least bigger. So you can see what a standard microarray data set might be. I don't know if they have made it uniform in terms of the data set. I'm guessing not. So I would guess that if you download different data sets, you will actually might get different formats. But basically, they have the name of your various different genes in your first different columns, identifications of them. So they may or may not be actual, contig 36292 is an identification for a particular gene, which may or may not have a name put in here or not yet. The actual data that we want to take a look at is between, if you can see that there's a black bar right here, there's this black bar here, and there's a black bar over here. So R, S, and T all refer to a single patient. This is open data, so showing it is okay, but if you do work with human data, you do have to strip things and make sure you can actually share it. So don't just automatically share human data unless it is open. This was open on the web. Anyone can download it, so it's safe to open up. But basically, if I take a look at, let me spread out R, S, and T across here so we can just take a look at. So this would be one data point. So before our data points were horizontal, now our data points are vertical. So the vector that I'd be interested in is actually this vector in this column right here. And it's the middle column. It's this middle column that you're actually interested in right here. In that middle column, we can see that this is a patient. We can see the age of the patient, whether or not they had this gene here or not. And so there's sometimes information about the particular patient up here. So if I clicked on here, that's basically where that information is in there. What we want is each of our components is that log10 ratio of patient data to control data. And so the log10 ratio of patient data to control data is given in this column right here. And so that is the data that you want to actually see. And if I go along here, you can see that, I probably shouldn't do that to the people who are online. I think I just messed up your eyesight. It goes up to 2400. So there's this is a very long vector, but that is the vector that you would want to analyze. Well, sort of. You do want to analyze that vector, but and for topological data analysis, probably taking that entire vector might be okay. But over here, let me actually go back up. We have R was the log10 intensities. So by the intensities, you have different amounts of DNA binding to your microarray. So sometimes lots bind, sometimes little binds, sometimes there is, so how well your microarray works, the higher the intensity, the more likely it's a valid result. If it's lower intensity, well then not very much of either, either your control or your tumor DNA or RNA, very little of it bound. And so if very little bound, maybe that's not very valid. And so a low log10 value means that your data is not very reliable. Versus if I take a look at, so a large negative number means not reliable in terms of the data. Versus if I take a look at the log10 ratio, remember we wanted the data to be, we took the top 1,500 most varying genes. Well, by varying, if I have something that is like 10 squared, then I've got, that the tumor DNA has been upregulated. So we've got much more of the tumor DNA or RNA, depending upon what we're measuring here. And so, a large number does tell me that it's varying, and so that's good. But 10 to the minus 2 means it's been strongly downregulated. So in this case, the absolute value, the higher it is in terms of the absolute value, the better off you are. Versus when you're close to 0, that means they're basically pretty much the same, so you're probably not getting to, you may not be interested in that information. It's probably just making, that's where we had the 1, 2, 3, 1, 1, 1, 1, 1. If you've got the stuff where everything is, the same, it won't all be the same ones, but it'll all be the, pretty much. If you basically have a bunch of zeros, you don't really need to compare a bunch of zeros to a bunch of other zeros. So that's pretty much how to look at it. And then the p-value tells you how significant the result is as well. So you can also take a look at that column to decide, is it maybe the intensity is not that great, but the variance is good, so maybe you want it anyway. Or maybe the variance isn't very good, but the intensity is high, and so maybe that's what you want to take. So the p-value does give you an indication as to whether or not you are interested in taking this particular value or whether you want to toss it out of your data. With topological data analysis, again, we're pretty immune to noise, but not completely, of course. So that would be something that one would want to play around with when inputting the data. I have not yet transferred this data into a format that Mapper will accept. We do have one of our classroom participants is working on that, so I'm hoping he gets to it before I do. But if we do, we might go ahead and see if we can take this one data set and see about getting it into a variety of different formats so we can play around with a single data set into different formats. But you're also welcome to, of course, play around with your own data sets if you like. So any questions in our remaining minute of class, or 2 minutes of class? Any questions? Okay. Well, then I think that will do it for today. If you've got additional questions on Mapper, please let me know. Hopefully we can continue the discussion offline as well.